Amelia Nasrallah, Center for Neuroscience here at St. Louis University. Last year, we were all together in a huge room, and who would have known that we're doing this virtually. So I hope that everyone is doing well and your families are also well. We are certainly going through some incredible times, but despite these many challenges, we are keeping up our spirit, our work, our dedication to the community and to science. We have a really interesting and nice symposium today. And although it's going to be done by Zoom, I'm sure that we will all enjoy it, have fun, and of course, we will learn a lot. So I'm going to just spend a couple of times presenting a couple of slides, and then Anya will take it from here, and then we'll come back and I will announce some of the winners of the presentation, and you'll hear some of the presentation from the winners, and then we'll end up with some of the talks from our postdoctoral fellows. So despite the challenging time, we had a good turnout, about 67 people registered. We've had 18 abstracts. Representation was good from St. Louis University, WashU, the University of Missouri, Providence College. We also have participation from Indonesia, Florida Atlantic University, Libya, and Boston. So when you look at the department, again, very broad, which is of course very, very nice. Representation from a variety of departments, including biology, biomedical engineering, communication sciences and disorders, internal medicine, molecular microbiology and immunology, neurology, neurosurgery, pharmacology and physiology, and psychology. So nice representation. Thank you everybody for doing this. If we look at the abstract and the representation of these abstracts, we had a broad topic areas that span from behavioral neuroscience, cognitive neuroscience, invertebrate neuroscience, neuropsychology, neurodegenerative disease, and pain. And when you look here at these pie charts, you'll see that although we have a lot of the graduate students that have presented, we also have had some from the undergraduate students. We hope that next year this is going to be increased. We certainly had a lot of undergrads presenting last year, but under the circumstances, this is very, very good. And 3 postdoctoral fellows that have also participated. But what's really nice is that if you look at the representation among the basic science and the clinical research, we are kind of closing this gap and narrowing the basic science and the clinical research. And of course, we want to see much more of that in going forward. So I really want to thank many, many people. First, Dr. Miller from the Scripps Institute, thank you so much for accepting our invitation, and the organizing committee that have done a fantastic job, from Anya and Fatma. These are our graduate students, Angie that has helped with the coordination, Andrew Butler, and that also has helped spearhead this effort. And of course, also all the students and postdoctoral fellow committee. So, you know, challenging times, but just an amazing job. It hasn't been easy. This is our first virtual symposium. So I'm sure that, well, I know that they had a lot of, we had a lot of little issues to dissect out, but we came out victorious. And of course, the judges, a number of judges that spent time reviewing the posters and selecting the winners. So I thank everybody for your commitment to the center and for making this a very successful virtual Neuro Day. And we hope that next year we will have this conversation in person. So without further ado, I will turn this over to Anya.
Thank you so much, Dr. Salamini, and yes, thank you all for coming to this talk and to this event. And so I would just like to take a few minutes here to introduce our keynote speaker, Dr. Courtney Miller, who is a professor in the departments of molecular medicine and neuroscience at the Scripps Research Institute. So thank you very much for being here today and accepting our invitation. I am going to take a few minutes to tell you about Dr. Miller's impressive career that spans over 2 decades. Dr. Miller completed her undergraduate degree at University of California, Santa Barbara, majoring in biopsychology. After graduating in 1999, she started her PhD in neurobiology at behavior at University of California, Irvine, under the mentorship of Dr. John Marshall, where she studied the biological basis of drug addiction in rodent models. After completing her PhD in 2005, Dr. Miller moved to Alabama, where she began a postdoctoral fellowship at the University of Alabama, Birmingham, where she studied neuroepigenetics. Also, while at UAB, she completed a degree in technology ventures at their School of Business in 2008. In 2009, Dr. Miller was appointed as an assistant professorship at the Scripps Research Institute in Jupiter, Florida. There in 2013, she was granted tenure, and actually just 2 weeks ago, she was promoted to full professor, so congratulations. Currently, the primary focus of Dr. Miller's lab is the discovery and development of therapeutics for diseases and disorders of the brain. Dr. Miller's lab is currently working towards treatments for substance use relapse, post-traumatic stress disorder, and glioblastoma. Dr. Miller's efforts have been recognized in a variety of ways, and I will certainly not name all of them or we would be here all day. But those include being selected as a fellow in the Kauffman Foundation Venture Capital Program. She was named Scripps Outstanding Mentor of the Year and was awarded the prestigious Presidential Early Career Award for Scientists and Engineers by President Obama. Dr. Miller also has a passion for advancing diversity in STEM and is a strong advocate for women in science. She co-founded the Professional Women's Nexus, PWN, in 2007, which is an organization driven by the mission of retaining women and other underrepresented groups in the higher ranks of science. I am so honored to have you here today, Dr. Miller. And please, I know we're all on mute, but just, please help me give her a warm welcome, and I will give it to her. And the title of her keynote talk today is Developing a Therapeutic to Prevent Relapse in Substance Use Disorders.
Thanks, Anya. Let's see if I can get my screens arranged here. Just bear with me for 1 second. Okay. Share that, and almost there. Okay, hopefully you can see that. Almost there, one more step. Okay. Great. So, it's a little tricky when you have these all set up because it fills both of my screens. So then if you can suddenly not hear me anymore, I won't see anyone motioning at me. I could just go on. So thank you guys so much for the invitation to present to you. As I was telling Anya and Fatma earlier, I really enjoyed the hour that I spent talking with some of the students. And yes, I think we could have gone on for a while longer. But let's get into it. Please, and I know you guys are on mute, but please feel free to interrupt me at any point. I really enjoy talking to a live audience, so I find the virtual format a little bit awkward. So dive in as if we were, in person. Okay, so I'm going to talk today about the work that my lab has been doing in collaboration with kind of a large team to develop a therapeutic to help prevent relapse to substance use disorder. Because of the nature of the work that my lab does, I get emails from people suffering from substance use disorder as well as PTSD. And there was one individual that emailed me that had a really articulate way of describing sort of how he suffers and a better way of putting what we do and why we do it in my lab. And he said that I could share his words. And so he says that I may never use meth again. I hope I don't, with all my heart. But all the therapy and meetings and love and support in the world does not stop the memories, and memories trigger use. So really the focus of our approach is to see if we can target those memories that are able to induce craving and motivation to seek drugs, even when a person is abstinent. So focus on the idea that memory drives relapse. So in my lab overall, we're largely focused on memory. And this has taken us into two directions, as I mentioned, PTSD and substance use disorder. For the PTSD work, we largely focus on epigenetics and neural circuits, but today we'll talk about substance use disorder, which really focuses us in on a molecular motor ATPase, myosin. And for that, we've moved into the area of medications development, looking for a treatment for substance use disorder. But that's also moved us into the area of oncology, which I could not have anticipated, as well as other drug discovery efforts, looking at another oncology indication, as well as infectious diseases, malaria in particular, and taking this back into studying these myosins in learning and memory. Okay, so what we have is this overall hypothesis driving our research that different types of memories can employ different mechanisms for their storage. Memory is typically studied in terms of how it's initially formed, or say long-term memory, which is defined as mechanisms involved in the 24-hour retention of a memory. But we're really interested in the long-term storage of memories and how that's achieved by the brain. And we have this hypothesis that there might be unique mechanisms for each type of memory. And that could allow a therapeutic approach that would enable their selective targeting. So if you could find molecular targets that are unique to, say, a memory associated with a drug of abuse, but not a memory that it would be considered adaptive, you might be able to develop a therapy around that. So we're starting off with understanding that substance use disorder is a chronic relapsing disorder and with limited treatment options. And broadly speaking, there are three types of relapse triggers: a small amount of the drug itself, stress, and then the memories that I mentioned. Memories are really a particularly challenge because they can have a wide range of representations that are associated with drug use. So examples of triggers can be music, chewing gum, seeing a snow-capped mountain because it can look like a pile of cocaine to someone, as well as money. So there can be some really abstract associations that make targeting them a challenge. So it's also a problem because exposure as brief as 33 milliseconds to a reminder of drug use can trigger the brain's go circuit, leading to that craving and motivation to seek drug, which is difficult to then put the brakes on. But on top of that, particularly salient memories can last a lifetime. And that's how the brain is designed. That's how memory is supposed to work. But memories associated with drugs are really salient. And so that can represent a lifelong risk of relapse. And so that's why we're interested in targeting those memories. So how to maintain abstinence? Well, in the case of many drugs of abuse, particularly stimulants like methamphetamine or cocaine, the only current option are behavioral modification therapies. Whereas for, say, like nicotine or opioids, there are replacement therapies available. For stimulants, there's nothing like that. So what we envision is behavioral modification plus a pharmacotherapy that perhaps selectively targets these drug-associated memories. Now, we're all really familiar with the opioid crisis, but unfortunately, it's a little bit like whack-a-mole. So in the world of drug use and abuse, sort of as one gets addressed, another pops up right on its heels. And that's what's going on with methamphetamine right now. So in 2017, which at this point are dated statistics, but it's the most recent of what's available, there were nearly 1 million people that were diagnosed with a meth use disorder in the US. And these two images here show the real toll that methamphetamine can take on a person. If you can see, I don't know if the font's too small, but these pictures are taken 2.5 years apart. And there are pictures that were taken at the time of this woman's arrest. And you can see the effect that methamphetamine has had on her, just her physical appearance. It's a really powerful drug. So as I mentioned, meth use is rising dramatically on the heels of the opioid pandemic, but it's also problematic because it's more pure and less expensive than during the previous boom. So you can see sort of in this graph here from the DEA showing over time the increase in purity with a drop in cost coinciding. And in fact, the majority of methamphetamine is no longer made sort of Breaking Bad style in home labs. Now it's largely being produced in countries like Mexico and Asia on a large scale, and then being brought into the US. Again, very inexpensive. And so consistent with the rise in use, as well as the increase in purity, overdoses associated with stimulants, including methamphetamine, rose by 25% from 2018 to 19, whereas opioids are a little more flatlined. They only rose by 9%, and actually they're starting to drop now. So again, unlike opioids, there's no way to reverse the effects of an acute meth overdose, really contributing to the accidental overdose deaths. The current standard of care, which I kind of touched on in the previous slide, because there are no medications available, it's strictly behavioral modification therapies. And unfortunately, it's just not enough. Relapse rates are reported to be as high as 90% in the first year of initiating abstinence, and a person can relapse on average 5 times in their lifetime before maintaining that abstinence. So the goal of our research has been to reduce relapse through the selective disruption of drug-associated memories. So we start out with the notion that drug associations or memories can trigger use, and that these can be weakened or quote unquote erased. And the typical approach has been to focus on accelerating the extinction or blocking reconsolidation of these memories. So if you're not familiar with extinction, this is sort of the repeated exposure to something without consequences. And so in the clinic, it's referred to as exposure therapy. And then reconsolidation is this period of time shortly after a memory is retrieved, in which the memory is labile at sort of a molecular level where it can be disrupted. And actually, back when I was a graduate student, we found that memories associated with cocaine can be disrupted through a blockade of reconsolidation. But focusing on these approaches of extinction and reconsolidation introduces a new problem. And that's that both of the strategies rely on active retrieval of the memory, but unfortunately drug users can have hundreds of triggers, and this includes unconscious motivators. So another example of this comes from the same person that I quoted earlier, and he says that all of my compulsions come from my thoughts trailing to memories of that feeling, the fun time, certain music, seeing a particular place, the smell of someone's cigarette, or even drinking a Diet Coke. There's no predicting the everyday experience that will be the next to remind me of the way it feels to be high. So the idea that you could sort of actively retrieve and target each of these memories in a clinical setting is not necessarily feasible. And that's how clinical trials or clinical studies are beginning to, the results are beginning to show. So an approach would be to devise a retrieval independent strategy and target memories when they're sitting in storage. But of course, this introduces a new problem. We have retrieval dependent strategies because they allow the selective activation of a memory of interest so that you can manipulate just that memory and not all. So that introduces the question of how can you achieve selectivity when a memory is just sitting in storage like a whole host of other memories. It would require identifying a mechanism that I touched on in the beginning with our overall hypothesis, identifying a mechanism that's unique to the storage of drug-associated memories. And that's what we stumbled across in the case of methamphetamine. So for this work, we've focused on dendritic spines because of their role in memory. So when a memory forms, new spines are formed, as well as there's an enlargement of existing spines. And this form of structural plasticity supports the functional plasticity, which is an increase in synaptic efficacy that occurs through things like insertion of more AMPA receptors that supports a strengthening of specific memories, specific connections to support memories. So this change in structural plasticity can support the maintenance of the functional plasticity of these memories. So we asked a simple question of what regulates structural plasticity. And this takes you to actin, which serves as a structural scaffold of spines. And I don't know if you can see my pointer or not, probably not, but I like this electron micrograph shown in the bottom left of the images. That turquoise teal color represents actin. And so I think it demonstrates nicely just how densely expressed actin is in the postsynapse. You can really envision it being a structural scaffold that drives the physical change in the shape of spines. Now, for some of the experiments I'm going to show you, it's important to understand how actin works to drive structural plasticity. So it works through actin polymerization, where actin monomers are added onto the plus or the barbed end of actin filaments and then removed from the minus end. And so the process of addition and removal from each of the ends is referred to as treadmilling. some of the experiments, we use latronculin A or LAT-A, which is a naturally occurring toxin made by the red sea sponge. And the way that it works is it sequesters actin monomers, or G-actin. So if they're sequestered and caged by latronculin, they're no longer available to be added back onto the F-actin. And so if it continues to be removed from the minus end of actin filaments, then it's going to be sequestered and no longer available for the addition, and the filaments will eventually fall apart. So that's how latronculin can achieve depolymerization. So we and others have shown that actin polymerization is required for memory formation. And here we found that it's required for the initial formation of a fear memory, but not its expression. So if you infuse latronculin at the time of conditioning directly into the basolateral amygdala, you can see that there is a deficit in learning when we test 24 hours later. And this was when the infusion is given 15 minutes prior to training. But if we change that and infuse latronculin prior to testing, the memory remains intact. And so it's telling us that pre-test infusions have no effect on a fear memory, whereas pre-training infusions do. So why could this be? Well, if actin is sequestering these monomers and the F-actin is no longer treadmilling, then the sequestration of available monomers will have no effect on the polymers. And this is this sort of stabilization of actin filaments occurs through the recruitment of actin capping and stabilizing proteins. So the idea is that at the time of training, actin is actively cycling, but by the time testing comes around 24 hours later, filaments have stabilized, and this happens within minutes of a training event. So latronculin has no effect. So then moving into our question of substance use disorder and drug-associated memories, we asked the question of how amygdala actin dynamics contribute to an established methamphetamine-associated memory. And to start off with, we use something really simple, conditioned place preference, where animals learn to associate a certain context. So on one side of the box or the apparatus with the availability of drug, and the other side of the saline, so that later if you put them back in a drug-free state and they're allowed to roam around, they should choose to spend more time in the context where they previously received drug. So we trained animals with methamphetamine over a period of 4 days, and then we gave them 2 days of sitting in their home cage. And then prior to testing, we gave them an infusion, again into the amygdala, just like with the fear memory experiment, of latronculin. Now, based on the fear memory experiment, we expected that the memory would be, that would remain intact, because we weren't manipulating it during the formation time when actin should be dynamic. And that's when we got our first surprise. So the way to look at this figure is if you focus in on the black lines, these are our control vehicle animals. And at test 1, which was the first day of testing, they showed a strong preference for the drug-paired side, which is labeled here as the CS+ for conditioned stimulus. And that preference for the drug-paired side persisted over 4 more drug-free test periods. So this is a really strong and lasting memory. Now, we expected that our latronculin animals would look similar, and if anything, they would maintain their memory longer over multiple testing periods, because we know that actin polymerization is also required for extinction, which is considered a new form of learning. But instead, you can see we got the exact opposite. So there was an immediate disruption of the memory, and it was long lasting. So at no subsequent test did the animals show a preference for the drug-paired side. So we started trying to figure out what in the world this meant. And we know that the first test is 15 minutes long. And so we reasoned that maybe, although it would be the opposite of what we would expect based on what we know about extinction and actin polymerization, we thought maybe there's really rapid extinction going on in the 15-minute period of that first test. And so it's showing up as no preference when you pool the full 15 minutes together. So for that, we took that first test and broke it up over 5-minute bins. So you can see that the vehicle-treated animals have a preference from the first 5 minutes that lasts throughout the test period. But this is absent in latronculin animals from the beginning, and again, persists throughout. So it's telling us that we have an immediate and lasting disruption of the meth-associated memory. So again, we went back to scratching our heads and trying to understand what was going on. And so we started to play sort of devil's advocate with ourselves and figure out what was going on. And one possibility is that it's a matter of memory strength. So maybe a memory that's associated with something fearful is disrupted by latronculin given at the time of testing, but not a memory for methamphetamine, simply because the fear memory is stronger. Now, that wouldn't fit with how we understand the strength of drug-associated memories to be, but it was possible. We were also interested in the possibility that the disruption of the meth memory was retrieval independent, which is not what you would expect, because that would mean that it was likely targeting it in storage. But we wanted to address that possibility by separating out drug administration and retrieval. So we wrapped both of these into a single experiment where we trained animals again for meth in CPP, and we did very strong training. So this should produce a stronger place preference in these animals. And we left the animals alone for 2 days in their home cage, and then we gave them an infusion of letrunculin just as we had done in the past, but this time we did it in their home cage. And we waited an entire day before testing them rather than doing the test 15 minutes later. So now retrieval and letrunculin are no longer paired. Yet we got the same results. So I'm showing the data in a slightly different format, but the animals that received vehicle showed a strong preference, and it was again absent in the letrunculin animals. So then we went back to figuring out, trying to understand what could this be. And there was another possibility that maybe there's something about actin dynamics that's unique to appetitive memories. So that's one difference between a fear memory and a meth memory. A fear memory is aversive, whereas a methamphetamine memory is appetitive. So we repeated the CPP experiment, but this time instead of giving them methamphetamine, we gave them Fruit Loops. Rats really like Fruit Loops. But we got another surprise. The Fruit Loop memory remained intact, just like a fear memory and unlike a meth memory. So this tells us that the disruption is independent of retrieval, and that it's selective for meth-associated memories, and it doesn't seem to be dependent on the memory strength. So from there, we decided to test it in a more sort of considered gold standard model of drug seeking using a form of self-administration to see if we got similar results. So for this, we use context-induced reinstatement of self-administration. So animals have a 2-week period where they self-administer methamphetamine in context A, and then a period after that where they're moved to context B and methamphetamine is withheld. Now these two contexts differ, as you can sort of see in the schematic, in the way they look, feel, and smell, so the animals can distinguish between the two of them. When they're moved to context B, the idea is to teach them that it's not the instrumental response of lever pressing that gives them access to methamphetamine, but instead it's the context. After extinction is achieved, the animals are given a day off, and then they get letrunculin, followed by a reinstatement test. And for this, they're placed back into context A. So the drug isn't available, but they're put back in the place where they previously had access to the drug, a little bit like in conditioned place preference. So this just shows their performance during training. There's an escalation over time that you would anticipate. And then when we move them to context B for extinction, initially they lever press even more because they're frustrated that lever isn't giving them methamphetamine. But they quickly learn that it's not going to and give up over that 10-day period. But if we return them back to their home cage, vehicle-treated animals reinstate nicely. Sorry, not their home cage, but context A. Vehicle-treated animals reinstate nicely. So they lever press about 50 times an hour when they're back in that environment where they previously had access to the drug. But this is significantly diminished in the letrunculin animals, and importantly, it persists into a test given the next day. So they have no additional access to methamphetamine between these two tests or letrunculin. So there's a single infusion that had a lasting impact. And in CPP, we saw that impact over 5 days. So what's going on? Well, we think that there may be something about methamphetamine that's interfering with the recruitment of these actin capping and stabilizing proteins, so that when letrunculin is introduced, it sequesters these monomers on actin that is still actively cycling, and this leads to the eventual sort of breakdown and loss of these filaments. Now recall, these are actin filaments that are supposed to be supporting the structural plasticity, maintaining a memory at the postsynapse. So to follow that up further, we asked whether or not the memory effects of actin depolymerization could be connected to changes in dendritic spine density if they really are supporting those spines. So for these experiments, previously everything I showed you was with rats. Now we've moved into using Thy1 GFPM mice. And they're really useful because they express Golgi-like fluorescence in several parts of the brain. They're typically used for their expression in the hippocampus, but we found that they have really nice sparse labeling in the basolateral amygdala as well. So because we'd only done this, observed this sort of phenomenon in rats so far, we wanted to make sure that it occurred in these mice as well. So for this, animals received saline during conditioned place preference or methamphetamine. And for the methamphetamine animals, of course, it's meth on one side, saline on the others. For the saline, it's saline paired with both sides of the apparatus. And then at the time of testing, animals either got vehicle or letrunculin. The saline groups are combined here because there was no difference between the vehicle and the lat A animals. But as we saw with rats, methamphetamine produced a place preference in these mice, and that was disrupted by latrunculin. Okay, but what about spine density? So methamphetamine we found induces a really dramatic increase in spine density in the BLA. This is a large change, but as you can also see, latrunculin returned that to essentially baseline. So this got us thinking about the clinical potential. So maybe we're onto something that could actually be useful, as sort of unexpected as it is. But obviously, actin would not be a good therapeutic target because it's critical for so many processes throughout the body. So for that, we turn to a form of myosin. Now, myosin is part of a huge superfamily, but there's one in particular that is of interest in the postsynapse. What brought us to myosin was focusing in on what's known about actin regulation in dendritic spines. And it's really complex. Not even everything is captured in this beautiful image. But if you zero in on this yellow circle, it shows myosin, and it's actually called non-muscle myosin 2, that regulates actin and its dynamics through a direct action on its physical movement in dendritic spines. So we reasoned, maybe this myosin could be safely targeted to achieve the same thing. And in 2010, I was part of a collaboration where we showed that non-muscle myosin and the 2B isoform of it does regulate actin dynamics during synaptic plasticity and memory formation. And for some of these studies, we used the small molecule inhibitor blebistatin. And what it does is bind near the ATP binding site, preventing the release and physical movement of this. It's hard not having the ability to just point at things like I would normally do in person, but it stops the motor head from moving on myosin, and that's what physically moves actin. So without the normal function of that ATP site, it can't move actin. So that's what blebistatin blocks. So as I said, we found through blebistatin and some genetic manipulations that non-muscle myosin 2B is important for memory formation in the hippocampus. And also in a follow-up study, we found that it's important in the basolateral amygdala for fear memory. So it suggested that we might be able to get at these meth-associated memories and their action in the amygdala. So now I'm going to give you a number of bullet point findings so that we can get to some more interesting stuff. So forgive me for this. But what we found was that intra-amygdala inhibition of non-muscle myosin 2 using blebistatin recapitulated all of the results that we got with direct actin depolymerization using latrunculin. And this was the case in male and female rats and mice, both adults and adolescents. We also extended our LAT-A findings to show that a single intra-BLA administration of bleb can disrupt drug seeking for at least 30 days. Previously, we'd only looked 2 days out, but you can see in the figure on the right that even a month later, after a single administration of blebistatin, there's a significant decrease in drug seeking. Now, in our vehicle animals, there's also a drop-off over time, but that's because we're repeatedly testing them without giving them any reinforcement with methamphetamine. So that what you're seeing is extinction. Really importantly, we also found that systemically administered bleb, so delivering an IP, is well tolerated, because again, that was our problem with actin as a target, is that it would not be safe to target with systemic administration, which is how, from a therapeutic perspective, you would want to deliver something. It was good luck for us that blebistatin is also 100% brain penetrant. This is often a very large barrier in working with small molecule inhibitors, is their ability to get through the blood-brain barrier, but it does very nicely. We also extended the selectivity of the effect to show that IP bleb reduces BLA spine density associated with meth-associated memory, so similar to latrunculin that's been infused directly into the amygdala, but that it has no effect on a fear memory. So this is all looking consistent. Okay, now we also confirmed the pharmacologic results, so using latrunculin and blebistatin, but with a genetic knockdown of the NM2B isoform, and that was done after training. So myosin 2, when we can knock it down with an shRNA against MyH10, and that's the 2B isoform. And the reason that we focused in on that was because of our prior data, but also because of its expression levels. So we had RNA-seq data as well as proteomics data from the basolateral amygdala, showing that MyH10 is really the predominant form of myosin expressed in the amygdala. Okay, so when we knocked it down, we had a disruption of the drug-associated memory, just as we had seen with small molecule inhibitors, and we also confirmed that there was a knockdown at the level of protein and that it did not have an effect on the other two myosin 2 isoforms. Now, this was pre-training knockdown because it was an AAV-based approach. So we also used siRNAs to ask a similar question, but where we would have more temporal control. So what we did was infuse the siRNA after training had been completed directly into the amygdala and looked at the effect on the place preference. And again, confirmed the expression. Okay, so so when you take all this together, we've seen a disruption in Sprague-Dawley rats, as well as 3 different strains of mice, using 5 different manipulations in 2 different behavioral models and 2 different labs. Because what I haven't mentioned is that NIDA also did a replication study, because what we'll move on to soon is a drug development project that NIDA has funded for us. But before doing so, because these results were so unexpected, that a single administration of a small molecule inhibitor could produce a seemingly long-lasting, if not permanent, disruption. And so if you could just excuse me for one moment, I'm sorry. Sorry about that. There are some challenges to COVID and working from home. My sweet 2nd grader just got home and he's having a tough time. And my husband, who's also a PI, is on another meeting downstairs on the other floor. So I'm so sorry about that. Yes, being a working mom has its challenges. Okay, so thank you. So NIDA was able to replicate our work, which was sort of a nail biter at the time because blebbistatin is extremely difficult to work with. But they had success with that, which led to funding of the application. Okay, so the effect of NM2 inhibition is surprisingly specific, we also found. So it produces an immediate retrieval-independent disruption of methamphetamine, and we also found amphetamine, which is essentially the same thing as Adderall. But interestingly, it didn't have this same retrieval-independent effect on spatial memory, a fear memory, one for food reward, or memories for other drugs of abuse. This includes morphine, nicotine, cocaine, or mephedrone, better known as bath salts, though the reconsolidation of several of these is also disrupted. I just need to do one thing. Sorry. These are unique things that I will remember forever. I actually gave my promotion talk a few months ago when my son and a pod of 2nd graders were doing school from our house virtually, and they were doing Taekwondo, pounding on the floor above me. It was interesting. So I swear I'm a very professional scientist. Okay, so we got a disruption of reconsolidation, but not this immediate retrieval-independent disruption. What we also found is that there's an interesting thing going on with polydrug use. So we were concerned, so the majority of people with substance use disorder are polydrug users. So if we're not getting this immediate disruption with, say, opioids or nicotine, what happens if we move into, an actual clinical setting and someone is also a smoker, for instance? Could it alter the actin dynamics in a way that methamphetamine is no longer, the methamphetamine-associated memories are no longer susceptible? And so we designed an experiment to test this, and we found actually the opposite. So whether meth is co-administered with nicotine or morphine or administered on alternating days, the NM2 inhibition still produces an immediate disruption, and it's similar to when methamphetamine alone is given. So right now we're doing studies that are self-administration based to see, but what it looks like is that it actually methamphetamine will confer susceptibility to memories associated with other drugs of abuse, which could be really promising. But on the level of specificity, we were also really surprised to find that it seemed to be unique to the basolateral amygdala. So we targeted other regions of the neural circuit that support these meth-associated memories. So specifically the dorsal hippocampus, nucleus accumbens, and orbital frontal cortex. And infusions of blood there had no effect. So there's something really unique about actin in the amygdala and amphetamines. Last, I would like to thank all the people on this page, but especially our funding, and most importantly, the people in my lab, past and present. We need an updated photo, but COVID has gotten in the way. So I've added some individuals on this side. Thank you so much for listening and for your patience with the working from home experience. I greatly appreciate it, and I would be happy to take any questions.
All right, well, thank you so much, Dr. Miller. Excellent talk, and certainly a great example of what happens in a time of COVID when our professional and family lives collide. So I know many of my own meetings have been interrupted either by, work in my building or my dogs. So I totally get it.
At least my cat didn't jump on my shoulder.
Right. So yeah, we'll take some questions for a few minutes. If you guys, if you have any questions, you can raise your hand and I'll try to get to some of you as well. Or I don't see any hands right now. So if anyone even wants to just unmute and go ahead, I'm fine with that as well.
I want to say hi to Anatosh.
Hi, Courtney, how are you?
Hi.
Great to see you. Fantastic talk.
Thank you.
I have a question. I can be in the line. If Daniela.
Go ahead. Yeah, go ahead.
Oh, you can go ahead, Anatosh. I'll follow up.
Okay, thank you. So yeah, it's a great talk, and I have always been like amazed with that specificity when I used to hear the talk back at Scripps, and this is really fantastic that it is only specific to methamphetamine and not the other type of drugs or memory. So from the biological point of view, to target this, it seems like it is still targeting actin or myosin, and there are the potential problems that you mentioned, which can be overcome by testing it specifically. But is there something that you think that methamphetamine increases this spine density, and with that some specific protein expression is methamphetamine dependent that binds to this complex of myosin and actin, which is not by the other drugs, so that protein can be targeted? It's a very up in the air question, but
no, it's not, Amitash. That's like half of one of my aims in the current R01, because we want to know the interacting partners. As I mentioned in my talk, we know almost nothing about this form of myosin in the brain. And there's a little bit from cell culture, but it's hard to draw connections because there's such specificity when you get into the brain and subregions. So that's what we're working on right now, among other things. We also did RNA-Seq to try to understand just what changes may occur at the transcriptional level with repeated meth administration versus cocaine in the BLA versus the hippocampus. And some interesting players came out of that, that there's some evidence for specificity between those two brain regions and between those two drugs, and for myosin and not in the other case, so to speak. And that was kind of what I was alluding to, that it's just too early to show it because it's really messy, but I think there's going to be something upstream of myosin, at the receptor level, for instance, that meth hits that cocaine doesn't, and that's expressed in the BLA and not in the hippocampus.
That's great. Thank you.
Very good. Dr. Salvemini, go ahead.
Yeah, that was a fantastic talk. Thank you very much. So my question were similar to Anutosh's in that these compounds are supposed to attenuate dendritic spine formation density.
Yes.
And so, I was impressed by the selectivity, because in other disorders, such as, for example, opioids and even cocaine, I think, there are alteration in dendritic spine morphogenesis, which are very much driven and dependent upon Rho GTPases such as RAC1.
Absolutely.
And so from that perspective, the uniqueness of the approach on amphetamine is fascinating in its own right. The other question that I have, though, is, myosin is also regulated by RAC1, and there are RAC1 inhibitors. Do they show efficacy in relapse to amphetamine? And do they also show the selectivity towards amphetamine and not opioids, for example?
Yeah, really interesting question. So what it seems to come down to is timing. So I follow everything that you're asking. In all cases with these drugs, every drug of abuse is going to change spine density, drive increases, right? As you're saying with opioids, cocaine, methamphetamine does the same thing. Now, I would expect levastatin to interfere with that formation for any of those drugs of abuse as it does in memory. But what's weird about methamphetamine is that myosin fails to, I believe, be dephosphorylated after the stimulus is gone when it's methamphetamine, but it does for the others. So it remains active so that later on when you deliver the inhibitor long after the drug is gone, it breaks down those, it stops myosin's activity, which breaks down the filaments. So we took a similar approach to what you're asking about with RAC1, but by focusing on ROC, because we thought perhaps because it has both phosphorylation and phosphatase activity, we could get at it that way. Because really, ideally for us years ago, if there was something that was already FDA approved or close, because there's a ROC inhibitor that's approved in Japan, maybe that would be the way to go about it. But when you're targeting the memory days later, everything else has already done its job and gone. The only thing that seems to be active is the myosin itself. So RAC has already done its job, ROC has already done its job, so inhibiting it later doesn't, unfortunately, achieve that effect.
Very good. So I believe Dr. Garg, you're next.
Oh, yeah. Very nice talk, Dr. Miller. I just had a very general question related to, sorry, my video isn't working for some reason, related to your new compound SR421.
Yes.
Have you seen any sort of effects on other tissues or general health, for example, on skeletal muscle or pulmonary function with that drug?
So we've done a fair amount of analysis of that. We'll have more data a few months from now, but the short answer is no. But remember, we're talking about single administration. So it's possible that chronic administration would. So we're talking about single administration with something that's very short acting. I didn't go into it, but the half-life is about 3 hours. So it's there for a very short amount of time. Now with skeletal myosin, we definitely don't see any toxicity. And I think in order to inhibit skeletal myosin based on some other data that we have with other compounds, you'd have to achieve a really high level or % inhibition of muscle, which requires a very high dose. So we don't see effects there. Things like blood pressure, heart function all seem to be fine.
Okay, thank you.
Yeah.
Great. So I think we have time for maybe 1 or 2 more questions. I don't see any hands raised, but if you do have a question, go ahead and ask. There's
question. So when you get to the clinical trials, when will this drug be used? Like, are you going to give this prior to a therapy session to try and bring up the memory and change what they're remembering? Is that the idea?
No, it's not, hopefully.
Okay.
That would be sort of a backup, but the idea, at least based on the animal work, is that you don't need that retrieval event, and that's
Okay.
what makes it appealing.
I see what you're saying.
Yeah, so what we'd like to do is say now, I've had a lot of conversations with folks at NIDA and clinicians, and so especially with COVID, I mean, it's getting harder and harder for anyone to go into an inpatient treatment program. So I don't know exactly how this will play out, but let's say ideal world, someone would be admitted into an inpatient program, abstinence would be initiated, and within, say, the first week, at some point in that first week, they would receive an IV infusion of this compound at night to limit the amnestic effects. And then, they would continue on with the rest of their sort of abstinence support treatment.
I see. Okay. So you figure that they'll only need this treatment like that one time?
That's what our data suggests. Of course, it's
Okay.
not in people, and so we won't know until we can get there. But yes, that's the idea, and I think that's what has us most excited, and
Sure.
why myosin is feasible to go after, because you're not talking about something really chronic.
Right. Thank you.
Yeah.
Great. Any other questions?
Thanks so much, guys. I really appreciate your time.
Thank you, Dr. Miller. Really great talk. Okay, so if there aren't any other questions, we can move on. So part of this event too, we would like to, we are going to award some awards for the abstract submissions, and I'll give it to Fatma and Dr. Salvamini for this.
Okay, so I'm very excited to announce the winners. We'll start with the undergrads. And the winner is Ali Abid from the Department of Biology. Well done. The graduate students, we have a tie, and the winners are Fatma Mufti and Katie Braden from the Department of Pharmacology and Physiology. Well done. And the winner of the postdoc is Dr. Rosato from the Department of Biology. Well done, Martina. So well done, everybody.
Excellent.
You'll get a plaque whenever we can get to those, and a little bit of a monetary award. So very well done, and thank you, judges, for judging the sessions.
Excellent. Thank you, Dr. Salvamini. So we will take a few minutes to show. So we had all of our abstract submissions. We had the presenters actually submit videos, very short videos, about 3 minutes long. So we would like to show the winner videos to you all. So we'll start with the undergrad. And give me 1 second here.
Hi, my name is Awais Paracha. I'm working with Ali Abid and Dr. Dixie Meyer on our project on opiate addiction and ACEs.
The screen is not shared.
Almost 2 million Americans struggle with some sort of opiate addiction. And, just as of 2017, we lost about 64,000 individuals to opiate overdose. And, our project examines the relationship between that and childhood trauma. And, in 2018, 3.5 million children were investigated for abuse. And, adverse childhood experiences like parental substance abuse, emotional neglect, physical and sexual abuse were all linked to opiate dependence. So again, our project wants to examine the relationship between opiate use disorder and adverse childhood experience.
All right. The rationale behind this study is to examine existing literature regarding opiate use disorder and ACEs in order to shed light onto relationships and correlations that may already exist between the variables as suggested by previous research. We want to expand on that relationship to determine if certain cases of ACEs are more linked to opiate use disorder or other key characteristics of opiate use disorder. This study hopes to allow for development of interventions in order to prevent opioid use disorder in the future. For the methods, a comprehensive literature search was performed on opiate use disorder and related terms. The search was then combined with another search locating ACEs literature. After completing search queries, the study initially identified 21,089 records. Of those, 20,436 remained after duplicates. 20,228 were excluded for not containing relevant keywords, leaving 208 after the screening process. 82 articles were excluded for being case studies and conceptual studies, leaving 126 meeting the eligibility criteria and being used in the qualitative synthesis. This study hopes to encourage new treatments and screening process for opiate addiction. If the findings suggest that trauma plays a role in opiate addiction, then physicians may use trauma to screen patients for a developing opiate addiction. This could help alleviate or diminish the current opioid crisis because physicians can use better screening methods and therefore be more careful when prescribing pain medications to at-risk individuals with past trauma like adverse childhood experiences.
Okay.
Politicians realize.
All right. So next will be our very own Fatma.
My name is Fatma Mufti, and I will be discussing the connection between CART peptide and sphingolipids in neuropathic pain. Neuropathic pain arises from a lesion or disease of the sensory nervous system, and it's estimated to affect up to 25% of chronic pain patients. So far, not much is known about the underlying mechanisms. Our lab has identified a couple of receptors that seem to be involved in neuropathic pain. One of them is GPR160 and its ligand CART peptide. The other is S1PR1 and its ligand S1P. Just to note, S1P in the body can be made either by salvage pathway or the de novo pathway. Considering that both CART peptide signaling via GPR160 and S1P play a role in neuropathic pain, we wanted to investigate if there is a connection between the two. Injecting CART peptide intrathecally in naive animals and using von Frey filaments to assess hypersensitivity, we see that the animals develop mechanical hypersensitivity. Thus, we wanted to examine the effect of administering sphingolipid enzyme inhibitors in the development of this CART-induced mechanical hypersensitivity. First, we inhibited serine palmitoyl transferase. And here we can see that the group of animals that received just CART developed mechanical hypersensitivity, as indicated by the reduction in the power withdrawal threshold. However, the group that received palmitoyl transferase inhibitor did not develop CART-induced mechanical hypersensitivity. Next, we blocked ceramide synthase. And here we see a similar trend where the group that received ceramide synthase inhibitor did not develop CART-induced mechanical hypersensitivity. Then we inhibited sphingomyelinase. Looking at the graph, we could see that the group that received the sphingomyelinase inhibitor did develop hypersensitivity, since the trend is similar to the group that received just CART, which indicates that blocking sphingomyelinase did not prevent CART-induced hypersensitivity from developing. All of this taken together, it seems that CART peptide-induced mechanical hypersensitivity involves the sphingolipids pathway in the spinal cord, and specifically the de novo pathway. With that, I would like to thank my mentor, Dr. Salvamini, our lab members, funding sources, and you for your time. Hello, my name is...
Very good. Congratulations again, Fatma. And the last one we'll see, let's look at this. I'm actually having trouble finding the last one. Give me 1 second here. Fatma, do you remember what Katie's
It's the TLR7. I see it. It's, yep, this one.
All right.
My name is Catherine Braden. I'm excited to share my research with you today, where we're looking at the role of TLR7 in oxysterol GPR183-induced hypersensitivity. This will follow up on a recent paper we published showing that GPR183 as a potential therapeutic target for neuropathic pain. In this paper, we found that nerve injury leads to upregulation of GPR183 in the spinal cord, leading to pain. We also found that direct activation of GPR183 using its oxysterol ligand induced pain in naive animals. And both of these effects were blocked using SA14, a novel GPR183 antagonist that we developed. So this indicates that GPR183 signaling in the spinal cord is contributing to pain states, but how is unknown. So to investigate these mechanisms, we repeated our previous experiment using SA14 given intrathecally in mice or its vehicle at 30 minutes prior to the oxysterol or its vehicle. And as you can see, the oxysterol produces this hypersensitivity or decrease in the polyphenol threshold, which SA14 is able to block. To look at the mechanism of how this is happening, we took the dorsal-horn spinal cord at 2 hours post-injection and ran RNA sequencing to determine what pathways were differentially regulated by GPR183 activation or inhibition. And as you can see in this heat map, there were a number of genes that were significantly upregulated in the oxysterol group in this red color compared to vehicle in this blue color. And SA14 was able to ameliorate a number of these genes being upregulated. We look at the gene ontology of what pathways were associated with these genes. We see a lot of toll-like receptor signaling and innate immune responses, which makes sense when we consider that GPR183's primary known function is in the immune system. We look a little bit closer at these genes. We see that 206 were significantly upregulated by the oxysterol, 84 were significantly downregulated by the SA14, and 14 genes overlapped between these groups, indicating that they're differentially regulated by GPR183. Of these, there was only one toll-like receptor, TLR7, and several other toll-like receptor-related signaling mediators. TLR7 is an endosomal toll-like receptor, and there's some previous literature to indicate that it may have a link with GPR183. In dendritic cells, it was found that GPR183 was a negative regulator of the type 1 interferon production that's downstream of TLR7 activation. They also showed that TLR7 activation was able to increase GPR183 mRNA. This previous literature supports our RNA-seq data and gives us a better understanding of what may be happening in the spinal cord. But of course, upregulation of a gene doesn't mean it's functionally involved. So to determine whether TLR7 can be functionally important to GPR183's ability to induce pain, we used a TLR7 specific inhibitory oligonucleotide, IR661, to see if TLR7 inhibition is sufficient to block OHC-induced allodynia. Indeed, our preliminary results show that TLR7 inhibition is able to dose-dependently delay oxysterol-induced allodynia, suggesting that TLR7 is functionally linked to GPR183 signaling in the context of pain. It is possible, since IR661 is unable to completely block oxysterol-induced pain, that other compensatory mechanisms are involved, and those will be investigated in future studies. With that, I'd like to thank my mentor, our lab members, our collaborators, our funding, and you for your time. Thank you.
Excellent. So those were our winners, and then I will hand it off to Fatma. We will actually hear from Martina, who was our postdoc winner directly today during her talk.
Thank you, Anya. Well, it's my pleasure to introduce our first postdoc speaker, which is Dr. Martina Rosato. She did her PhD in the Molecular and Cellular Neurobiology Department at the Center of Neurogenomics and Cognitive Research in Amsterdam, the Netherlands, investigating the biological functions of schizophrenia risk genes. She's currently a postdoc in Dr. Zhu's lab in the Department of Biology at SLU. Her current work is focused on investigating the roles of chemical and electrical synapses in the brain and neural development. Thank you for joining us, Dr. Rosato, and we're excited to hear about your great work.
Thank you. Let me share the screen. Okay, can you all see it, right?
Yes.
Okay, so we'll talk about a project we have on the transcriptome analysis on Limnes stagnalis. Limnes stagnalis is an invertebrate, a snail, and it's very important in neuroscience because it has a very simple central nervous system. It has around 20,000 neurons, which are organized in a ring of connected ganglia. So here are all the ganglia connected. And very importantly, those neurons are easily identifiable. So that means that we can recognize each single, well, pretty much each single neurons. And this is important for the study, for example, of neuronal networks. So in an example here with the respiratory network of the limnia, they could actually identify the exact neurons that were involved in this response with the respiratory. And also those neurons we can study, so we can have primary culture from the neurons and study them. So we can study learning and memory and synapse formation and characterization. So here are two examples from like also the respiratory neuron. We have the left pedal D1 and the VD4. So we can actually identify those neurons and easily manipulate them. Now, despite its importance, there's very limited characterization of the genome and the transcriptome. So in our lab, we did for the first time a transcriptome from different developmental ages. And we got young, immature snails, adult snails, and old snails. We extracted the central nervous system and we performed transcriptome analysis on the mRNA from the central nervous system. And when we look at the data, we could see that there's age-dependent separation of our transcriptome. So if we look at principal component analysis, we can actually see, so we look at the principal component analysis, we can actually see we have a first component with has this around 70% of contribution, and it actually identifies the young CNS transcriptome, while the second principal component is actually distinguishing between adult and old transcriptome. This data is further confirmed. So if we look at the most differential genes, we can see that the pattern of expression of these genes is strikingly different in the central nervous system from young snail compared to adult and old. Now, due to time, I'm not really going to dig too much into the genes. So I'm only getting a few examples of genes that are involved, for example, in synaptic plasticity or synapses. So one of the first example is the molluscal insulin peptide. And this insulin peptide has been shown to be important for long-term memory formation in limnia. So what they did is they were looking at the appetitive behavior, which is basically when the snail opens and sort of like munches. So open and closes their mouth. And what it happens is that they were doing a conditioned taste aversion. So when you give a solution on when you drop a little bit of a solution of a sucrose solution, which is palatable for the snail on their lips, they actually have this appetitive behavior. On the other hand, with a stressor solution like KCl, this appetitive behavior is inhibited. So in the conditioned taste aversion, they actually give the snail both the palatable, so the sucrose and the KCl solution in a row. And that means that the snail is conditioned to associate a palatable stimulus with something that is not nice. So their appetitive behavior is inhibited. And this conditioned taste aversion is actually, it lasts. So one day after, they still remembers this conditioned taste aversion. And this is where we have this long-term memory formation. And they saw that during this memory formation, they have an increase in insulin peptide expression. On the other hand, when they were giving an antibody against this insulin peptide, they could still have the conditioned taste aversion, but then long-term memory formation was not there anymore. So one day after, the snail couldn't remember anymore this association. Next, when we look at some synaptic genes, first of all, I want to remember all of you that we have both chemical synapses and electrical synapses. So we have more than one type of synapses in our brain. And then when we look at chemical synapses, we have an action potential in our presynaptic neurons, which lead to the increase of intracellular calcium, which then lead to the synaptic vesicle docking and fusion on the membrane, and then the release of neurotransmitters. And we have receptors, of course, on the postsynaptic neurons. In the electrical synapses, instead, our action potential is directly transmitted to the postsynaptic through these gap junction channels, which lead to the direct communication between the cytosol of the two neurons. When we look a little bit in deep into these electrical synapses, so those gap junctions are formed by proteins which are called connexins in vertebrate and innexins in invertebrate. And in our lab, we were actually the first one to clone the innexins genes in Limnia stagnalis. And then we were looking for these genes in our transcriptome. And we could see 3 of those genes were expressed in our transcriptome. And interesting, they have differential expression in the different age, so in young, adult, or old CNS. So this seems to suggest that different ages have different expression of innexins, and therefore their gap junction might be slightly different. Furthermore, we actually validated this information with qPCR. So we could validate that this differential expression was actually there in our central nervous system. And we're following up on this innexin. On the other synapse instead, when we look at the canonical synapse, so we have this docking effusion of the synaptic vesicle, and we have different proteins which are involved, and one of these is complexin, which binds to the snare proteins, and therefore, together with the snare proteins, it's important for the priming and diffusion of this vesicle. Now, importantly, those vesicles are usually released through this triggering from calcium, but sometimes some of these vesicles can just spontaneously fuse to the membrane. And what they saw both in mammalian, so mostly mouse and rat, but also in invertebrate, in Drosophila melanogaster, the fruit fly, and in the C. elegans, they saw that the complexin was important for both the calcium trigger release and also the spontaneous release. Also, we also did clone the complexin in limnia, and we saw that especially the snare binding domain is highly conserved. And furthermore, what we also saw is that when we look at the phylogenetic tree of the complexin, so we can see that when we look at the vertebrate, we have two different complexin proteins, complexin 1 and 2, which are of course very similar. While in our invertebrate, we only have one protein. And this is further confirmed by Western blot. So we have an antibody which specifically recognizes our limnia complexin. And when we look at the mouse complexins or the limnia complexin, of course, this antibody can recognize both the complexin 1 and 2 in vertebrates. So we can see here 2 bands, and in our limnia, we only see 1. When we get to our actual transcriptome, then we can see that, again, we have a differential expression of this, of the mRNA of the complexin in the different ages. So this is also something that we're very interested into exploring. And this is where we get to our actual future studies. So we have Brittany Holscher in our lab. She's developing the CRISPR-Cas9 technique, where she's actually developing this for the innexin genes. So the idea is to knock down the innexin genes and in our embryos, in the snail embryo. And then we can look at the, both behavioral, but also we can extract the brain from those snails and look at the neurons. So we could, for example, extract the neurons and then look at the immunofluorescence and see, well, in this case, we're looking at 2 neurons that are actually doing chemical synapses, but we can actually also extract neurons that are known to form chemical synapses, electrical synapses, and therefore look at whether their synaptic connectivity is still there. And we can also look at the electrophysiology. So as I was saying, we can easily distinguish which neurons we are extracting. So we can extract neurons that are known to form chemical synapses or neurons that are known to form electrical synapses. And with this CRISPR technique, we can either knock down our innexins, which are important for the electrical synapses, or we can knock down the complexin, which is important for the chemical synapses. And we can look at the electrophysiological pairing of the neurons. And in the chemical synapses is interesting because we can see, of course, the triggered release of neurotransmitters, but we can also see the spontaneous release. And of course, then we can also look at the development. And I think I'm already a little bit, up of my time. So with this, I will just conclude this with my acknowledgement. Of course, all of the XU Lab, especially Dr. Zhu and Brittany and Chidera, and also Dr. Lin for helping us, and our grant fundings. Thank you. And I will stop sharing. So I don't know if you have any questions or anything.
Great work, thank you. Any questions? If you have a question, feel free to just unmute yourself and ask it.
Thank you.
I guess I have a question. So do you think that the 2 complexins that are in mammals or in mice specifically are going to have similar functions to what you're seeing in the snails?
Yeah, that's a good question. So in general, yes. So they're all involved in the synaptic vesicle docking. Although, well, this is still not investigated in our snail, but they saw that, for example, one difference in the mammalian and the invertebrate like Drosophila and C. elegans complexin was that in the mammalian they had the same effect when you were knocking down the complexin. They had the same decrease in evoked and spontaneous release of vesicles, while in invertebrate, at least in the Drosophila and in the C. elegans, it seems to have an opposite effect. So it seems that the knockdown is actually increasing the spontaneous release. So that's something that might be also interesting to see whether in our snail we have something similar to what we already saw in the invertebrate.
Awesome. Well, if there are no other further questions, we could go ahead and transition to our next speaker, which is Dr. Silvia Squillace. She has worked on inflammatory and chronic pain during both her master's and PhD. Her PhD was in pharmacology and toxicology at Piacenza, University of Rome. Currently, she is a postdoc in the Salvamini's lab studying how chemotherapy affects cognitive functions in collaboration with Dr. Farr's lab, both labs at the Department of Pharmacology and Physiology at SLO. Dr. Squillace, thank you for joining us, and we're excited to hear about your great work.
Thank you, Fatma, for the introduction, and thank you for inviting me today. So I'm gonna share the screen with you. All right. You all see my screen? Perfect. All right. So thank you all for attending this session this afternoon. And today I would like to show you some data from one of our latest projects, which is investigating possible involvement of S1P1 and TLR4 receptor in cisplatin-induced cognitive impairments. So currently in the United States alone, we have more than 15 million cancer survivors, and this population is expecting to grow up to 26 million in the next 20 years. And this is undoubtedly a remarkable achievement. However, this growing population has started to show some severe side effects and long-lasting side effects due to chemotherapy neurotoxicity. And in particular, 50% of patients reported a decline in cognitive functions. And this decline affects multiple aspects of cognition, such as memory, attention, learning, executive functions, and processing speed, generating a state that is commonly known as chemobrain or chemofog. Molecular mechanism underlying chemotherapy-induced cognitive impairments, or CICI, are still mostly unknown. However, several studies are focusing on neuroinflammatory pathways that are triggered by chemotherapy administration, in particular in the prefrontal cortex and in the hippocampus, that are two brain regions involved in cognitive functions. However, today, no FDA-approved drugs are available for treating or preventing CICI, which means that this is a huge unmet clinical need. So our lab and others over the last 2 decades have intensively studied sphingosine 1-phosphate signaling at the S1P1 receptor, demonstrating that the activation of these receptors, which in the central nervous system is expressed increased neuronal excitability and an increased reactive gliosis. So the activation of S1P1 in the central nervous system lead to neuroinflammatory state. FTY720, which is clinically known as fingolimod, is an S1P1 functional antagonist, and it has been approved by FDA for the treatment of relapsing remitting multiple sclerosis, which means that its safety profile is well defined and well known. FTY720 has been shown to prevent and reverse other neurotoxic effects of chemotherapy, such as peripheral neuropathy, thanks to its ability to dampen central neuroinflammation. And another important characteristic of this compound is its anti-cancer activity. And this is particularly important considering the possibility of co-administering this drug with chemotherapeutics. So our working hypothesis is to verify if the co-administration of FTY-720 with chemotherapy can actually preserve cognitive functions. This is just a brief overview of our experimental protocol. So for this experiment, we use both male and female mice wild type. And these are we had 3 experimental groups. So we have the vehicle group, the animal treated with cisplatin, and the animal that received both FTY-720 and cisplatin. And in this case, FTY-720 was injected 15 minutes before cisplatin administration. So after the treatment phase, animals are allowed to rest for a couple of weeks. And then for the following 4 or 6 weeks, we evaluate different aspects of cognition through different behavioral tests. And these are some of the results that we obtained. So this is the T-maze. This is a test that evaluates learning and memory retention. In this test, the mouse is placed in a maze, and then a buzzer sounds and the door opens, and the mouse is free to move in the maze. And after 5 seconds, a mild shock is applied to the maze. So mice have to learn how to reach this safe platform here within 5 seconds in order to avoid the shock. And we have a training phase or acquisition phase on day 0. And then on day 7, we have a retention phase in which we evaluate what they learned and what they remember. So these 2 graphs here are referred to day 7. And as you can see, both males and female mice that receive cisplatin, they failed more times in reaching the platform in comparison to the vehicle. Whereas the animals that also received FTY, their performances are completely comparable with the controls. Then we have novel object recognition. In this test, 2 similar objects are placed in a maze, and the mouse is free to explore the object for 5 minutes on day 1. And then the following day, one of the 2 objects is removed from the maze, and a different object is not. So theoretically, mice should be more attracted by the novel object because they should remember the old one. So these 2 graphs here are referred to day 2, and on the y-axis we have this index that expresses how much mice spent with the novel object, exploring the novel object. So as you can see, the male mice that receive cisplatin, their values are all around 0, which means that they spend approximately the same amount of time exploring the 2 objects. Whereas both controls and animals that received FTY, they definitely spend more time exploring the novel object. So this is valid for males, but actually in females, we did not see a real deficit in this test, because all mice in all group explore definitely more the novel object. A kind of opposite situation we observed in the puzzle box. So this test evaluates problem solving and executive functions, and it consists in a light chamber and a dark chamber connected by a tunnel. Mice generally are attracted by a dark environment. So in front of the tunnel, we put different obstacles of increasing difficulty as the days go by. And the animals have to learn how to remove the obstacle in order to reach the dark chamber. And so as you can see, male mice in this test did not develop a real deficit, whereas the female mice that received cisplatin, especially in the last 2 days, they struggled definitely more in comparison to control to figuring out how to remove the obstacle and reach the dark chamber. Whereas the females that received FTY here in blue, their performances were definitely better. So at this point, since it is not known how chemotherapy trigger S1P, S1P1 signaling, we hypothesize a possible involvement of TLR4 receptor. So, and these hypothesis is validated from some data that are present in literature. So cisplatin is known to bind the TLR4 receptor, and actually we are performing some docking experiments as well in collaboration with Dr. Aknat. The activation of TLR4 receptor has been shown to is linked to learning and memory impairment, and the activation of TLR4 also induce an hyperactivation of some key enzymes that are involved in S1P production. And finally, TLR4 and S1P1 downstream signaling pathways, they are found to cooperate to increase inflammatory responses. So given this background, our hypothesis was to verify if in the absence of TLR4, we could preserve cognitive functions in mice after cisplatin administration. So first data that we obtained came from the analysis of samples of hippocampus and prefrontal cortex. And what we found is that in these two brain areas, TLR4 is overexpressed after cisplatin treatment. And we also decided to invite to send some samples to Dr. Spiegel at the Virginia Commonwealth University for some sphingolipidomic analysis and trying to consolidate our hypothesis of a link between TLR4 and sphingosine signaling. And this is our experimental protocol for this experiment. So it's pretty much the same that I showed you before. In this case, we use a wild type and TLR4 knockout mice. And I'm going to show you just the result that so far were more interesting, the one in T-maze. And as you can see, as expected, both males and females that received cisplatin, again, their performances were definitely worse in comparison to controls. Here in females, probably we did not reach significance in this experiment because of the low N value. So we need to repeat this because these are very, very preliminary data. However, even with just 4 mice, you can see how both males and females TLR4 knockouts, they do not develop any kind of deficit in this test, and this is pretty encouraging. So to conclude, what we observe is that cisplatin impairs different aspects of cognition in males and females. That FTY-720 given concurrently with cisplatin prevents CICI in both sexes. Cisplatin is able to induce an overexpression of TLR4 into brain regions that are critically involved in cognition, which are the prefrontal cortex and the hippocampus. And TLR4 knockout mice receiving cisplatin do not develop CICI. So collectively, these data suggest S1PR1 and TLR4 as promising targets to prevent chemotherapy neurotoxic effects on cognitive functions. I would like to thank my mentor, Dr. Daniela Salveni, and all the people in our lab, especially Dr. Doyle, who performed the Westimulate. I would like also to thank Dr. Susan Farr and her lab. They are teaching me everything about cognition, which is a new field for me, and it's very interesting and exciting for me. I would like to thank also our collaborator, Dr. Aknat and Dr. Spiegel, and I would like to thank also our funding, and thank you all for your attention.
Wonderful, thank you. Any questions?
I have a question. This is Dr. Sampson. All right. Sorry. A very nice talk. What's the pharmacological profile of this blocker? Do you see what would be possible side effects of this blocker?
This blocker, well, FTY-720 is as a well-defined safety profile, and it has already been used in clinic, and actually also regarding some other chemotherapy side effects like peripheral neuropathy, it is advancing in clinical trial. So its safety profile is pretty well defined, and it has been shown that it only induces mild bradycardia, but it's perfectly, I mean, it's just in a small fraction of patients, so it's considered safe, pretty safe. And so it's well known and well defined.
I have another question for you. It's very nice work. I would assume then that an NF-kappa B inhibitor would block the effects.
So this is a good question because actually, so the cartoon that you saw is a simplified version of what it could happen. So for sure, everything, I mean, we are hypothesizing that these two pathways that converge in the activation of NF-kB could be probably the main trigger in this process. But of course, this is a very complex phenomenon, and so probably yes, but I cannot be 100% sure of that because of multiple pathways that for sure are involved in this, and so for sure there are other regulatory genes that are involved and other genes that are expressed that could affect cognition as well.
Thank you. Very nice presentation.
Thank you.
Do we have any other questions?
I have just a really basic question. It's a really nice talk. How long, because I don't know much about this topic, how long does the cognitive impairment last? Is it permanent?
So in a patient, actually, it could last for several months and sometimes also years. So it's, yeah, it's definitely long lasting. In mice, in our experiment, we test up to 3 months. Oh no, sorry, maybe 2 months after the last chemo injection. And then we sacrifice them to collect tissues. So we never go beyond 8 weeks of testing. However, yes, it's definitely long lasting. And so it's something that must be addressed as soon as possible.
Yeah, absolutely. Thank you.
Thank you.
Hi. Yeah, I also agree. It was a great talk. I'd like to follow up on that question. Do we know anything about return to baseline? Is you know, so the deficit lasts for a period of time, and there's a return to baseline. And is baseline or worse than pre-deficit?
So yeah, that's what I was I was discussing with Dr. Miller. So I know from patients that this can last for several months and years, and in some cases, then it tends to disappear over time. So in patients, after several months, some of them at least, they return to normal cognitive levels. In mice, we personally haven't tested for how much it lasts. This deficit lasts. For sure, they are very, very long lasting. But since we are focusing with our project on understanding which one could be the mechanism of onset, we are trying to prevent them or try to short them as much as possible. So we personally did not investigate for how much they last.
All right. Well, thank you so much. Again, nice talk.
Thank you.
Wonderful. Well, thank you everyone for tuning in. Thank you to all of our speakers. And now I will hand it back to Anya for closing remarks.
Yeah, thank you, Fatma. So just very briefly, I speak on behalf of Fatma and myself. Thank you all, first of all, for attending. I know this was kind of a weird format. We had to do, this was definitely a learning curve for both Fatma and I and the rest of the organizing committee, but I guess now we can say that we know how to organize a virtual event, so that's great. Again, Dr. Miller, excellent talk. It was great talking with you. We're grateful for your time. We'd also like to thank all of our postdoctoral fellows for the presentations, Dr. Rosato and Dr. Scolace. Also to all the undergrads and graduate students and postdocs who submitted abstracts, great abstracts and great videos. And of course, they, instead of creating the posters, they had to do video submissions. So I know that was new for a lot of us as well. A special thanks to Angie Coleman for her invaluable help and guidance during this organization of this event. I don't know how she does it. I don't know how, I just don't get it, but she just seems like she has 1,000 hours in a day. So thank you so much, Angie. Thank you, Dr. Salvamini and Dr. Butler, who have helped us at all, as well. And just in general for, this event is supposed to be organized specifically by the trainees, and what a great opportunity for us to be able to do this on our own with the guidance of the center. Finally, thank you to all the judges. I don't even know, thank you to everybody. And hopefully next year we will be able to meet in person. So let's see what happens. Yeah, I don't know if anyone, Dr. Butler or Dr. Salvamini, want to say anything, but after the conclusion of this event, we would like for the center members to stay on and just have a few minutes kind of on a little bit of a social time with Dr. Miller as well. Thank you.
So I'd like to say really hats off to Fatma and Anya for organizing this amazing work and the great learnings that you guys have done and experienced. And of course, Angie, who's phenomenal as well. So thank you, Dr. Miller, for giving such an outstanding presentation. I've learned a lot. It was wonderful. And thank you for everyone to be here today and support this virtual Zoom meeting that I personally think went very well despite the circumstances. But again, hats off, Fatma and Anya, and congratulations to all the winners. Great job. Thank you again. And we're looking forward to next year in person, okay? So.
Yeah, I think that went as well as it could have, Fatma, so.
Yes.